Hello friends, welcome to Microbiology Insights. So today we are going to discuss about microorganisms or microbes. So what are microbes? Microbes are basically living organisms which can be unified based on a single property that all of them has a size of in the micrometer range. What do you mean by that? 1 micrometer is equal to 10 to the power minus 6 meter. Means if you take a 1 meter scale and divide it into 10 lakh times, then each unit is 1 micrometer. So we can well understand that how much smaller it is. That's why to visualize a microbe, you need a special kind of instrument, which is microscope. We all know that microscopes are a combination of lenses, and as lenses magnify objects, so using microscope you can visualize that much smaller organisms. So microbes or microorganisms are of different types. So what are the types of microorganisms? So microbes includes bacteria, it includes fungi, which are of two types basically. One is yeast and another is mold. It includes microalgae, it includes protozoas, it also includes another group of organisms which we can't define as that. In that sense, it is not that kind of true organisms, but another very much important pathogenic organism, that is virus. So to summarize, what are the different types of microbes? Bacteria, fungi, protozoa, microalgae, virus. So coming to the cellular form of microbes. So when we talk about microbes, the first microbe that comes into our mind is bacteria. So bacteria is basically a cellular type. It has a cell type which is prokaryotic. So living cells could be distinguished on the basis of the presence of a particular membrane-bound organelles. Those cells which has those membrane-bound organelles are called eukaryotes, which includes the nucleus, and there are organisms which do not have any membrane-bound organelles, that are the prokaryotes. So bacteria are basically prokaryotes, means they do not have any membrane-bound organelle like Golgis, like mitochondria, like endoplasmic reticulums, and most importantly, the one which basically distinguishes a prokaryote and a eukaryote is the nucleus. Means prokaryotes do not have a membrane-bound nucleus, means nuclear membrane-bound nucleus, while eukaryotes, they have membrane-bound organelles as well as the membrane-bound nucleus. Now coming to the cellular type, what do you mean by that? Means microbes, they may be unicellular or they may be multicellular. But among the unicellular ones, you have microbes which are prokaryotic as well as which are eukaryotic. Means they are unicellular forms. Bacteria are basically unicellular prokaryotes, while yeasts, we all know about yeast, they are the type of fungi which basically participates or which are responsible for bread making. We all know that they are main ingredients of bread making. They are eukaryotes, but unicellular eukaryotes. So to differentiate between a prokaryotic cell and eukaryotic cell, we take a model organism as bacteria in case of prokaryotes and yeasts as eukaryotic unicellular organism. There was a scientist, there was a plant ecologist, American plant ecologist, Robert H. Whitaker. So Robert H. Whitaker basically taxonomically he differentiated living organism into five kingdoms. So when we talk about taxonomy, we know that taxonomy is basically grouping organisms. So there are certain hierarchies which are basically followed while classifying organisms. What are they? The biggest group is the domain, followed by subgroup the kingdom, followed by the phylum, followed by the class, followed by the order, family, genus and species. So species is the unit of taxonomy. So Robert H. Whittaker, what he did, he basically classified total living organisms into five kingdoms. What are they? They are the Monera, the Protista, fungi, Animalia and plant. Microbes are included in the in three groups of these three kingdoms rather, where the Monera, the Protista and the fungi. Though fungi also have macroorganisms or multicellular bigger organisms which we do not need microscope to look at. So bacteria are included in the Monera group. The Protista group includes two types of organisms. One is called the animal-like protist and another is called the plant-like protist. The plant-like protist includes the microalgae. While the animal-like protist, they includes the protozoa. The major example of protozoa we all know that it is Plasmodium, the one which is responsible for malaria. Then you have Leishmania, which is responsible for colors or etc. So this is the taxonomic position of bacteria. However, Robert H. Whittaker didn't include virus in its particular classification system because the basis of Robert H. Whittaker's kingdom classification system was, one of the basis was cellular type, because viruses do not have any cellular kind of morphology. So basically it was not included in the five kingdom classification as proposed by Robert H. Whittaker. Now, to define or to explain the characteristics of bacteria, bacteria is diverse, is hugely diverse. Diverse with respect to shape, diverse with respect to its oxygen requirement, diverse with respect to its mode of nutrition, diverse with respect to its gram character, and this is a very important thing which I'll just explain a few minutes later. And also it is diverse with respect to metabolic activities, means there are numerous microbes that can degrade numerous types of substances. They participate in various activities which are important for ecologically. So basically, when we differentiate bacteria or classify bacteria on the basis of shape, there are different types of shape of bacteria. Some are spherical in shape, some are rod in shape. The spherical ones are called the coccus. You have subgroup of coccus. Then there is bacillus. Then there is a curved rod, which is the vibrio. Then you have spirillar form of bacteria. Then you have certain pleomorphic bacterial species, which basically, since they do not have any cell wall, they lack cell wall, so they takes a pleomorphic shape. So these are the various classifications of bacteria on the basis of their shape. And among the spherical ones, the one which is spherical, there are Diplococcus, the two spherical shaped bacteria means cells attached to each other. Then there is Streptococcus, where the spherical cells are attached in chain. Then there is Staphylococcus, where the spherical cells are arranged as the bunch of grapes. And there is Sarcina, where it is the spherical cells are arranged in cubes. Then you have Bacillus, you have chain of Bacillus, you have palisade cells, where the Bacillus cells are attached adjacent to each other to form a particular association. Okay, and you have the curved rods. in typical books you will see this term, it's comma shaped. Okay, so it's basically the curved rod. You have another group, this is called Coccobacilli. It's a mixture of coccus and it's an oval shaped structure. It's called Coccobacilli. One of the typical example of Coccobacilli is Azotobacter. You all know it is one of the free living nitrogen fixing bacteria. We will discuss it later about the nitrogen fixation and its importance. Then basically this is the classification of bacteria on the basis of shape. Coming to the gram character, a scientist called Hans Christian Gram in 1884 he basically devised a method of classifying bacteria into two groups, which is based on the cellular feature, that is the surface of the bacteria. That is the gram positive and the gram negative. It's a staining procedure. It's basically a coloring procedure where bacteria could be differentiated on the basis of the ability to take up different color differentially. So it's a differential staining process where you can classify gram positive and gram negative bacteria. Bacillus subtilis is a typical example of gram positive bacteria, while E. coli or Escherichia coli is basically an example of gram negative bacteria. So also microbes could be classified on the basis of their oxygen requirement. There are microbes which grows in presence of oxygen strictly, like us, and there are microbes which do not require oxygen, means oxygen is toxic to them. Those are called anaerobic bacteria, and those that grows in presence of oxygen like us are called aerobic bacteria. This is the major two class. Also have other classes which are called the facultative anaerobes. Escherichia coli is a facultative anaerobe which can switch between aerobic and anaerobic condition based on the availability of oxygen. There are aerotolerant anaerobes like Lactobacillus, the one which is responsible for curd making, we all know. Aerotolerant anaerobes. We have two other physiologically distinct groups that have different types of oxygen requirement. One of them are microaerophils or microaerophilic organisms, which requires a reduced oxygen tension or reduced oxygen availability for their growth or survival. Typical example of microaerophils are azospirillums, a group of nitrogen fixing bacteria. And the other physiological group that has a high requirement for carbon dioxide. They require about 5 to 10% carbon dioxide for their metabolism. One of the major examples of such organisms are Campylobacter jejuni. We all know Campylobacter jejuni are responsible for duodenal ulcers. These kind of organisms are called capnophiles. So capnophiles are those that require a high carbon dioxide availability for their growth. Bacteria can also be classified on the basis of their nutritional requirements. So based on the nutritional requirements, bacteria can be classified into 6 major groups. 2 of the groups are based on the requirement of carbon sources. 2 of the groups are based on the electron sources, and 2 of the groups are based on energy sources. So based on the carbon sources, the 2 groups are autotrophs or heterotrophs. Now the autotrophs are the one which basically utilizes free carbon dioxide as the sole source of carbon. It's much like the green plants. There are certain bacteria which are autotrophic, means they utilizes free carbon dioxide as their sole source of carbon. The other group that is based on the carbon sources are the heterotrophs, which basically depends on the preformed organic matter to gain their carbon. Coming to the next group, where bacteria are classified based on the electron sources. So basically here there are two groups. In one group, the electron donor is inorganic compounds, and in the other group, the electron donors are organic compounds. In case of those bacteria where the electron donors are inorganic compound, they are called lithotrophs. So litho means rocks. Rocks generally represents inorganic substances. So in that case, lithotrophs are the one whose electron donor for metabolism is are inorganic compounds, and there are another group, another member, that is the organotrophs. The organotrophs uses organic carbon as their electron source. The last two groups, they are classified based on the energy source. So based on the energy source, the two groups are the phototrophs and the chemotrophs. The phototrophs, as the name suggests, as we can well understand, the energy source is light, while the chemotrophs, the energy source is chemical energy. So there are different examples of these different types of groups, and these groups are also interlinked, like there are chemolithoautotrophs, photolithoautotrophs, chemoorganoheterotrophs, and like this means a single bacteria can have a carbon requirement that is fulfilled with the help of carbon dioxide. Its electron source is fulfilled from inorganic compounds, and the energy source is of course chemical. There are bacteria which uses energy, light as energy source, and so there are a mixture of this kind of nutritional requirements. So this is the major classification type, or the diversity of microbes or bacteria generally. Hope you have enjoyed this session, and please subscribe our channel and click the bell icon to get more video updates from Microbiology Insights.